We are Purdue GOGEC, and this is our work on the design of an engineered probiotic for the treatment of celiac disease. Up first, a little background on celiac disease. It's an autoimmune disorder which affects 1 in every 133 healthy average Americans, meaning that it's very prominent. Its symptoms are largely caused by the presence of gliadin fragments in the small intestine. As you can see, looking in the upper right, gliadin is one of the 2 subcomponents of gluten. The other is glutenin. Symptoms of celiac are also caused by genetic variations and mutations, particularly in the gene HLA-DQ, which is a receptor on antigen-presenting cells in the small intestine. Currently, for those individuals suffering from celiac disease, the only available treatment is adherence to a strictly gluten-free diet. But newly developed therapeutics are being explored with different targets to allow people with celiac to eat foods that they want to and live a more well-rounded life. Particularly is Coumax, which is an engineered metabolic enzyme that has been developed at the University of Washington, and it's pictured in the top right of this slide. Additionally are different synthetic antagonist molecules such as lorazepam, pictured on the bottom right. We decided to use 3G assembly for the creation of our plasmids because of its ease of workflow, as well as its standardization and ease of use with an already existing large library of parts. How 3G assembly works is you start off with your adapters, promoters, ribosome binding sites, genes that encode proteins, as well as terminators, which are shown up at the very top as those single symbols. And then you run a Golden Gate reaction using those to create what are called transcriptional units, which are a single sequence that encodes one gene. You can also design them to encode more than one gene, but in their simplest form, encode one gene that can then be PCR'd and amplified and stuck together using those overhangs, which here are shown as those colored boxes. And then a Gibson reaction can be run to place them in a larger plasmid backbone. This method is designed so that it can all be run within a single day, so it allows for a much faster and simpler method of plasmid assembly. Each of the gene parts that we designed, we used benchling for, and we designed them with 3G compatibility in mind. What this means is that primarily we ensured that each of the genes did not encode for any BSA1 cut sites, as that would cause the gene encoding protein to be cut off prematurely and not allow for our desired proteins of interest to be created. But additionally, we also had to add ending sequences on both ends of the gene so that they could be used in the standardized method of 3G assembly. For example, all of our protein encoding genes, they had to have C and D linkers on them so that they could be used in the standardized method. Additionally, all of our parts were codon optimized using IDT's codon optimization tool so that they could be effectively created in E. coli. And virtual Golden Gate and Gibson reactions were run using software Genius Prime to ensure the usability of our designed parts within the larger system. Shown here is the overall design concept of our engineered probiotic. In general, we wanted something that would react to gluten and then produce a protein to then break it down, specifically the subcomponent gliadin. What we found, however, was that there were not any promoters that directly sensed gluten, as gluten as a whole complex is a very large molecule and would not be getting into E. coli to interact with a promoter directly. So what we did instead was we pivoted to an iron sensing route. Most flour in the United States has iron in it, and flour is made of wheat, which contains gluten. So we decided to use that sort of peripheral method. In breaking up into two main parts, the first is iron sensing and lorazepatide secreting, and then lorazepatide acts to activate the second portion, the second set of circuits, which is lorazepatide sensing and serine protease releasing. Lorazepatide was chosen because it also has effects of lessening the symptoms of celiac disease. Additionally, there is a promoter that interacts directly with lorazepatide to be used in the second part of our project. Serine proteases were chosen as the class of protein of interest, particularly subtilisins or subtilisin-like serine proteases, because they cleave proteins in general, but gliadin in specific, in the region that interacts directly with one of the key regions of gliadin for causing the symptoms of celiac. So cutting gliadin in this region would effectively inhibit gliadin from being able to cause symptoms. For the iron sensing part of the project, we designed a genetic construct capable of sensing and responding to iron level. This was the FIR ferric uptake regulator. The FIR box within promoter regions mediates ferrosensitive transcription, and so it's a perfect promoter for the use within our system. Additionally, signal peptides were coded for because they can be attached to target peptide therapeutics to be then secreted out of bacteria. We use these signal peptides to secrete lorazepatide from E. coli. For the iron sensing construct, we put it together using 3G assembly, as detailed in the previous slide. You can see the agarose gel electrophoresis of a PCR product in the lower left images there, and you can see that it yielded several prominent bands. What this shows is that we got a good concentration and a relatively good purity of that transcriptional unit that was created. These bands were then later gel extracted and transformed into E. coli and plated on these plates as shown in the right. These were two different attempts of transformation of the transcriptional unit of that iron sensing construct. As you can see, the top plate did not yield any colonies, and the bottom plate did yield colonies, but there wasn't the phenotype visible on the agar plates. These colonies were supposed to produce M. scarlet, in which case they would have been red and visibly red to the naked eye on these plates. What this then led to of us doing a bunch of troubleshooting and kind of trial and error analysis, but we ultimately have not been able to find why the M. scarlet was not being produced in this case when it should have been based on our research. Pictured here is a fluorescence assay used with the iron sensing construct, and we measured the optical density at a wavelength of 700 nanometers and the M. scarlet fluorescence over a period of 24 hours using a plate reader. We varied the levels of EDTA from 0.1 millimolar to 8 millimolar. And for a little bit of background, EDTA is a chelator which is widely used to bind iron ions. The bottom left graph is a readout specifically of M. scarlet fluorescence from LB media grown with that transformed E. coli. And then on the right it shows the plate reader. The squares that are in yellow were grown in LB media, and the cyan were grown in M9 minimal media with increasing levels of EDTA concentration. And the controls are shown in the upper right of each of those images, with the yellow still being an LB and the M9 in cyan. All of the white backgrounded spots were wells that were left blank. Pictured here are the designs for the gene parts to be utilized in the second part of our engineered probiotic. The two on the left and the upper right are our serine proteases, or our proteins that are intended to cut gliadin. These are Coumax, subtilisin A, and subtilisin C. The colored bar in the center of each of those images is the gene portion, and the purple flanking tags on each end are the overlaps needed for 3G assembly. Highlighted in blue are the BSA1 cut sites, detailing that they are only present on the ends of each of those proteins, of each of those genes. The fourth picture here is the KR2HLAT linker sequence, which was developed based on a paper previously published by Hanka et al., and it is used in our constructs to be able to secrete these proteins out of E. coli and then cut using OMP-T in the KR2 sequence of this linker. Here are the gene circuit systems that were designed for the secretion of the serine protease proteins of interest, which again were Coumax, subtilisin A, and subtilisin C. All three of those are represented on here by the rectangle containing POI, that stands for protein of interest. And so all three of them could be used and tested within either circuit system. The top circuit system shows secretion using IL-22 HLA system, which is from the Murray lab, courtesy of Dr. Green. It is already designed as a 3G compatible part and for use as that. The top of those two circuits shows the POI connected to a precision site and then HOIA. Later, the HOIA would be cut off in post-processing using the precision enzyme. And the second circuit shows HOIB, HOID, and TOL-C, which are essentially helper proteins that are needed for the HOIA secretion system to get the protein of interest outside of the E. coli cell. HOIA mediated secretion is a system natively found in E. coli and has been used before for genetically engineered proteins and their secretion. The bottom set of circuits is secretion using a slightly different HOIA system with an OMP-T linker, which was adapted from a paper previously published by Hanka. And the main differences here between these and the ones above them is the linker sequence, the KR2 versus the precision. Additionally, the amount of HOIA included in this is much shorter, which is why it's known as HOIAT. It was just the ending tag sequence, as minimal of protein as needed to hopefully affect the protein of interest the least in its creation and post-processing. Additionally, in the second circuit of this bottom set, TOL-C is needed to be created, or OMP-T is needed to be created in order to cut at the KR2 linker site. The second method that we looked into was simply cell lysis. While this is not as preferable as secretion, it was another avenue to be pursued and explored if secretion was not going to work. In both of these schematics here, the POI is the serine protease, whichever one of the three could be plugged in that spot. And the top circuit shows a non-quorum sensing version. Both utilize the phage protein Phi-X174E. And then the bottom is a quorum sensing version of that circuit using SynR and then a promoter that reacts to SynR for our quorum sensing capability. In moving forward with this work, we would like to continue testing the larazotide secretion construct in order to get expression in response to iron content. We would also like to construct our serine protease constructs in vitro and compare the efficacy of our designated proteins of interest, the Coumax, subtilisin A, and subtilisin C, with both release mechanisms, the secretion versus the lysis. We also would like to combine larazotide and serine protease constructs to get our overall desired sensing behavior and move from a design to an actual prototype. Purdue iGEM would like to thank Dr. Leopold Green, Dr. Carrie Clace, Dr. Stephen Lindeman, and Dr. Mohit Verma for their guidance and support throughout the project. We would also like to thank the Purdue University College of Engineering and College of Agriculture for their generous gifts, as well as thanking the SURF and SCARF fellowships for the opportunities that these programs provided over the summer of 2022.